Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating and crucial microscopic structures in the human brain, the transmitter-gated channel. These tiny molecular machines are the fundamental chemical to electrical translators of our nervous system. Without them, our neurons would be completely isolated, unable to communicate, and all human thought, movement, and behavior would instantly cease. We will build our understanding from the ground up, starting with a simple analogy, moving through their biological structure, and finally uncovering the beautiful mathematics that govern how they work. Let us dive in. To begin, imagine you are standing in front of a massive, heavy vault door. This door has no handle, but it does have a very specific keyhole. No matter how hard you push or pull, the door will not budge unless you have the exact right key. In the brain, neurons are surrounded by a protective wall called the cell membrane. To send electrical signals, they need to let specific charged particles, called ions, inside. Transmitter-gated channels are exactly like those locked vault doors embedded in the cell membrane. The key in this scenario is a chemical messenger called a neurotransmitter, which is released by a neighboring neuron. Now that we have our analogy, let us look at the formal neurobiological terms. A transmitter-gated channel is a type of ligand-gated ion membrane receptor. It is a complex protein structure that spans entirely across the neuronal cell membrane, forming a central pore that is normally closed off to the outside environment. Unlike voltage-gated channels, which open or close in response to changes in the electrical membrane potential, these transmitter-gated channels are strictly controlled by chemical binding. The neurotransmitter acts as the ligand, floating across the synaptic gap and binding to a highly specialized receptor site on the extracellular side of the protein. So, what exactly happens when that chemical key enters the lock? When a specific neurotransmitter binds to the receptor site, it triggers a rapid conformational change. This means the protein structure physically shifts and changes its shape. This structural shift opens the central pore, suddenly making the membrane permeable to specific ions, such as sodium, potassium, calcium, or chloride. This is the critical moment of translation. The channel is acting as a primary molecular machine, taking a chemical signal from a presynaptic neuron and converting it into an electrical signal in the postsynaptic neuron by allowing charged ions to rush in or out. Before we move on to the mathematics, let us see this mechanism with a concrete visual example. On the screen, you are looking at a cross-section of the neuronal cell membrane. On the left, you see the channel in its resting, closed state. Notice the red neurotransmitter molecules floating in the extracellular space above. On the right, one of those neurotransmitters has successfully bound to the receptor site. You can clearly see the conformational change. The pore has widened. The blue arrows represent positively charged ions now freely flowing down through the open channel into the intracellular space. This simple physical opening is the basis of all neural communication. Having seen how they open, we must ask, why do these channels matter so much in our daily lives? Without transmitter-gated channels, the entire nervous system would instantly shut down. They provide the fastest way for neurons to communicate. Every single time you decide to move your arm, a neurotransmitter called acetylcholine binds to these channels on your muscle fibers, opening them to trigger a muscle contraction. If you touch a hot stove, the rapid withdrawal of your hand relies on these channels firing in milliseconds across multiple synapses in your spinal cord. Furthermore, many common anesthetics and life-saving drugs work specifically by targeting these channels to block pain signals. Now that we understand the biological structure and importance, let us look at the physics of the ions flowing through them. When a transmitter-gated channel opens, the ions do not just wander aimlessly into the cell. They move based on very specific physical rules. Their movement depends on two main factors. First, how easily they can pass through the open pore, and second, how strongly they are being pushed or pulled by electrical and chemical forces. To calculate the actual electrical current flowing through these open channels, we use a modified version of Ohm's law, which beautifully adapts basic electronics to the biology of the brain. This formula we just mentioned is the mathematical heart of transmitter-gated channels. The equation states that I-ion equals G-ion times the quantity Vm minus E-ion. Let us break down every symbol. I-ion represents the ionic current, the actual flow of electrical charge, measured in amperes. The symbol G-ion stands for conductance, measured in Siemens. Conductance tells us how many channels are open. It increases as more neurotransmitters bind. The term inside the parentheses, Vm minus E-ion, is the electrochemical driving force. It represents the net pressure pushing the ions. If this driving force increases, current increases. But if conductance drops to zero, current instantly becomes zero. Before we move on, let us see this with a concrete example involving an excitatory synapse, the brain's accelerator. Suppose glutamate opens sodium channels. We are given a conductance of 5 × 10⁻⁹ Siemens, a resting membrane potential of -65 millivolts, and a sodium equilibrium potential of +55 millivolts. We substitute these into our formula. Crucially, we multiply the voltage term by 10⁻³ to convert millivolts into standard volts. Subtracting 55 from -55 gives a driving force of -120 millivolts. Multiplying this by the conductance yields our final answer, -600 picoamperes. So we have calculated a current of -600 picoamperes. But what does this actually mean for the cell? In neurobiology, a negative current signifies an inward flow of positive charge. Because the inside of the cell is at -65 millivolts, there is a massive inward driving force pulling the positively charged sodium ions inside. When the channels open, a strong surge of sodium rushes into the neuron. This massive influx of positive charge will depolarize the cell, making the inside less negative. This pushes the neuron closer to firing its own action potential, successfully continuing the chain of communication. Now, let us examine the opposite scenario, an inhibitory synapse, which acts as the brain's braking system. Imagine the neurotransmitter GABA opens chloride channels. We are given a conductance of 2 × 10⁻⁹ Siemens. The neuron is slightly depolarized at -50 millivolts, and the chloride equilibrium potential is -65 millivolts. We apply the exact same formula. Notice the double negative when we subtract -65 from -50. This turns into a positive addition, giving a driving force of +15 millivolts. Multiplying by the conductance, we get a final current of +30 picoamps. Let us interpret this new result. We calculated a final current of +30 picoamperes. A positive current indicates an outward flow of positive charge. However, mathematically and physically, an outward flow of positive charge is exactly equivalent to an inward flow of negative charge. Physically, this means that negatively charged chloride ions are rushing into the cell. Because the current membrane potential was less negative than the equilibrium potential, the electrochemical pressure pushed the negative ions inside. This makes the inside of the neuron more negative, effectively inhibiting it and preventing it from firing an action potential. We have covered a lot of ground today, so let us summarize the key takeaways. First, transmitter-gated channels are the essential chemical to electrical translators of the nervous system. They remain tightly closed until a specific neurotransmitter binds to them, acting like a precise key in a lock. Second, once open, they allow specific ions to rush across the cell membrane, and this flow is strictly driven by the electrochemical forces we calculate using Ohm's law. Whether they let in sodium to excite the cell or chloride to inhibit it, these channels are the fundamental reason neurons can communicate rapidly to produce all human behavior.